was nice. We were spread out for the other one because we all sort of congregated. Yeah, I like that. He was good. I really thought he was excellent. So funny because there was we have like an email list distributor. You sort of stop where are you going after city council issues. You use it when you offset it starts getting hot. Where you the city council meetings are on because you watch it online. Somebody said something. I wrote back to him, but I told him how long were you in school at? He's being completely racist. And then I told him I consulted like 3 or 4 other people on the team on starting who were like founders. And I wrote a note saying, I expect you to write a note. It's a nice city though. You to retract that. I like it. Yeah. Publicly. A lot of cities should be on this list. Of course, he hasn't done that. Well, this is like.
Before I go see donors, but they're pretty paranoid here.
Yeah, I just thought since we were on the thing, I should. I'm not thinking. I've been.
Yeah. So I'm going to be given the thumbs up, so I'm just waiting for that. Thumb may be approaching. Thank you. So, welcome everybody to this public session. This is a little bit different than a number of the other presentations that you've been hearing. We are gathered here with a most august panel on the subject of can regenerative medicine turn back the clock on aging? A minor question. Before we get to the panel, I usually like to get a sense of the audience, so I'm going to take a quick survey here to sort of gauge the relevance to our topic. So who here is getting older? Okay. There are a few that, number of hands, those that said no, let's talk afterwards. We really would like to get the secret to your success. My name is Jonathan Thomas. I, for the past 11 years, have had the great privilege of serving as the chairman of the board for the California Institute for Regenerative Medicine, which is CIRM by acronym, which you may have heard that name bandied about here over the course of the conference. More on that in a bit. Want to introduce first to you our panelists who are going to be speaking after I do a short presentation. In order here to my right, first we have Dr. Deepak Srivastava, the president of the Gladstone Institutes here in San Francisco and professor at UCSF. Deepak's research focuses on understanding the causes of heart disease, one of the leading causes of death in the US, and identifying new and really interesting ways to treat it, some of which I'm sure he will regale us with shortly. To Deepak's right, Dr. Tom Carmichael is a neuroscientist and the chair of the Department of Neurology at UCLA. Tom is doing some fascinating research into developing ways to repair the damage caused by a stroke, the third leading cause of death disability in the US. And to his right, one of our premier patient advocates over many years now is Adrian Shapiro, who is a patient advocate for sickle cell disease, the co-founder of Access Advocacy, a patient support group, and a regenerative medicine activist, ensuring that the patients' voices will always be part of any discussion about research and clinical trials. So, before we get to their comments, our format here is, I will give a brief presentation to tell you a little bit more about CIRM and what we do and how it impacts the area of aging. And then we will then have each of the panelists in turn give a brief presentation and comments on topics of their particular interest. After that, I will ask some Q&A of our panel. And then last but most not least, if any of you have questions that you would like to ask of our panelists, we will have them at that time. So, on your screen here is our, actually the cover sheet of our many of our documents, our strategic plan, various other things. Real life is our motto at this point in time. Before we give any presentation, we always start with a look at our mission, which is accelerating world-class science to deliver transformative regenerative medicine treatments in an equitable manner to a diverse California and world. So, CIRM, as you may or may not know, is a creature of the odd process in California whereby you can qualify something for the state ballot if you have enough signatures to get it there, which is generally, give or take, about 1 million. And back in 2003, my predecessor, Bob Klein, came up with the idea of creating a state agency to fund stem cell research in California, and he, together with longtime counsel James Harrison and a number of others, crafted so-called Proposition 71, which he got the 1 million signatures for, qualified for the 2004 general election in November of that year, needed 50% plus one vote to pass, and it got 59, and CIRM was thereby created, which had this agency in place to fund stem cell research, and importantly, the measure provided for $3 billion in state general obligation bond money to fund that research, which immediately put California not just further on the map, because we had tremendous scientists in the state, but because we had the luxury of this funding, immediately made California the epicenter in the United States of stem cell research. As you can see from this particular slide, we are patient-centric. We are really, think of us as sort of the bank where we give grants out to projects in the stem cell space. As you can see, we have funding throughout the research continuum, starting with basic research through so-called translational research, which is sort of the period of time between basic research discoveries and the clinic, which we hope that the work ultimately ends up in so that we can test what we have come up with to see if it actually will bear fruit and can lead to therapies and cures for patients. We also have a very active infrastructure program, which is variously defined. In the early days when CIRM was just getting going, it was a lot of bricks and mortar. We have 12 stem cell institutes throughout the state of California that are either freestanding buildings that were built at the time, such as UCSF. Any of you who've ever been there, it's an architectural marvel on the cliffs up in the Parnassus campus. And if you are agoraphobic, I do not recommend walking on the external staircase, which ends up hundreds of yards over a cavernous fall, but it's quite something to see. Or we have these facilities that were redone to suit stem cell research, such as at UCLA, for example. In later years, infrastructure has been sort of defined to refer to various other things as well, one of which is something called the Alpha Stem Cell Clinic Network, which is, at the moment, 5 different, or 6, I should say, different research centers that have soup to nuts clinical trial facilities for stem cell projects that are either in phases 1, 2, or 3, and they have a number of things that they share which make this network a very special thing and really a one-of-a-kind network in the country. It has both CIRM-funded projects in clinical trials as well as non-CIRM-funded projects. So we go on to here. I've sort of gone through a bit of this. Creation in '04. We ran out of our money. We ran through our $3 billion in 2020. Not an easy thing to do. It's a lot of grants. And because of that, and because the field is sort of now in early to midlife, the need for continued research was never greater. So Bob Klein came back, ran Prop 14 in the November 2020 election. It got 51% of the vote, a little bit closer than the 59 in 2004, but nonetheless, it passed. And as we were fond of saying, we weren't in this for the A, we wanted the pass, and it did. And the impact on patients in California, nationally, and worldwide of it passing was monumental, as was the case with Prop 71 in 2004. Since we started, we've funded over 1,000 different cutting-edge and transformative programs. As I said, we have 80 first-in-human clinical trials covering every disease virtually you can think of, from the most common and pronounced as far as numbers of the population down to many of the most rare diseases that you have most likely never heard about unless you have family members or know of folks that are friends of yours who have those conditions. We have over 3,200 patients now enrolled in this Alpha Clinics network, which is a number that's steadily rising as the trials increase. So one of the things I wanted to tell you about was, just to give you an example of the power of regenerative medicine as it affects people with various conditions and how regenerative medicine can turn back the tide of aging to a certain extent, which is, of course, the topic of this discussion. And so on the screen, you see Anna Kuehl, who was an avid naturalist and hiker, and came down with age-related macular degeneration, which, as you may know, is the leading cause of vision loss in the elderly in the US, if not in the world, caused by the thinning of the part of the retina in the eye that is responsible for giving us clear vision. So we had a clinical trial with a company that we funded called Regenerative Patch Technologies. And on the right there, that little thing you see held by the tweezers is the patch. And it's a tiny scaffold that is seeded with stem cells and placed in the back of the retina. And immediately upon doing that, Anna reported an improvement in her vision. She was ultimately able to improve her reading ability on an eye chart by 17 letters, huge improvement, and is now once again able to go out and enjoy nature as she did before. Anna highlights the power and the potential of regenerative medicine to not just halt diseases, but to reverse them, which is the ultimate goal, the holy grail. But harnessing that power and realizing its potential is not an easy thing, and everybody, is by nature sort of impatient, but science takes time. If we're going to do it and do it right, it takes time. So we get now to our portfolio. 23% of our CIRM projects that we've funded are involved with age-related diseases. And you can see whether it's heart disease, stroke and dementia, vision loss, cancer, musculoskeletal diseases. That's a big chunk. Remember, we fund $3 billion, and by the time we're done with this next round, it'll be $8.5 billion. So multiply that by 23%, and you can see that this is a central focus, among others, that we have here at CIRM. And we've developed now a strategic plan that started about a year ago. We have every 5 years we come up with a new strategic plan that is keyed off of the state of the science in the field at the time the plan is developed, so as to keep us continually on the cutting edge of what needs to be done in research to further advance the field in that time frame. So we have our sort of 3 goals, which you see there are advance world-class science, when you can see, you can sort of read what that means. I don't need to recite it. Deliver real-world solutions, and very importantly, provide opportunity for all. One of the great underpinnings of CIRM is the notion of being able to have treatments available and affordable to all citizens of California. And towards that end, we have very recently established something called the Accessibility and Affordability Working Group, which is charged with how do you make these treatments, some of which are very expensive, available and accessible to all populations, particularly those in underserved communities that traditionally have had less access to health care than others. So we are very dedicated to that. We're very dedicated to diversity, equity, and inclusion, and have DEI as fundamental tenets of all our awards, both with respect to the teams that we fund and with respect to the projects and who has access to clinical trials and all that sort of thing. So when you kind of take it all as a single body of work, what has been enabled by CIRM is really quite something. And we have a number of members of our incredible team here today. Could everybody just stand up so you can be acknowledged? All of our CIRM folks. Katie, wave, please. Thank you. So with that, let's now move on to step 2 here, which is presentations by members of our panel. And we're going to start with Deepak. So thank you.
Thank you, John, and it's a pleasure to be here with you all today. What John mentioned is that the CIRM has focused on many diseases of aging because it's during the aging process that many of our organs degenerate. And what we'll talk today about is I'd like to talk about more in terms of health span rather than necessarily age span. Aging is often confused, studies on aging, with extending the actual lifespan of an individual, which I think is a bit of a different problem. But health span refers to during those years where you are alive, to be as healthy as possible, and regenerative medicine certainly has the possibility to do that. Now, at the Gladstone Institutes, where I work, we are largely focused on diseases of aging, and we're organized into highly collaborative teams of scientists focused on heart disease, brain diseases like Alzheimer's, Parkinson's, which clearly have an increased incidence as people age, viral diseases, and we've seen during COVID how it's the elderly that have really been the most susceptible to death from COVID, and then immunologic disorders where the immune system clearly deteriorates as we get older and is the basis for so many diseases as we're beginning to understand. Now, in all of these cases and many other diseases of aging, the problem that we boil down to is actually pretty simple. These are conditions where either cells that should do their normal function are now lost in our bodies with the age, or with age, they are now no longer functioning the way they should. So not everything falls into that, but most human disease actually falls into those two large categories. And it just so happens that some of the key breakthroughs in the last several years finally give us an opportunity to address those two issues at their root. Stem cell technologies obviously afford us the ability to replace damaged cells, and gene editing technologies, particularly CRISPR, offers us an opportunity to go in there, and if there's a code that's wrong that results a cell in misbehaving, then we can go in there and correct that code. At Gladstone, we have the good fortune of having two of the key inventors of both of those technologies under one roof within running labs at Gladstone, with Shinya Yamanaka, who invented iPS cells, and Jennifer Doudna, who invented the CRISPR technology. And so we're using those approaches across all those disease areas at the moment. My background is as a pediatric cardiologist, and I thought I'd share with you some of the work that we've done, largely with CIRM support, on addressing this issue of losing heart cells that results in heart failure, both in children as well as in adults. And there are currently 24 million people worldwide who suffer from heart failure, which is a term we use in cardiology to refer to a situation where the heart's still pumping, of course, but not well enough to support all the needs of the body. And people have a slow but steady decline in the setting of heart failure, ultimately to the point where they need a heart transplant to survive. And even in a country like the United States, where we have significant resources, there are only about 3,500 heart transplants done every year in the whole country. And there are many countries around the world where it's either not allowed or simply not done. And so those people simply die. And so we cannot continue to just accept that if you lose cells in that condition and so many others, that you just die. which is what we do currently. And I'm very hopeful that much of what you're hearing at the meeting this week, and as John mentioned, CIRM has funded a number of trials, is fundamentally going to change that. An approach that we took in my laboratory some years ago that was inspired by Shinya Yamanaka's discovery that you could reprogram cells from one state to another, in his case from a fibroblast or skin cell into a stem cell, led us to ask, could we regenerate heart muscle not by transplanting cells, which has promise but also has many challenges, but rather harnessing the potential of cells that are already in the organ to do something that they normally don't do, which is to beat. And it turns out that only half of the cells in each one of our hearts in this room are muscle that beats. And the other half of your cells in the heart are simply support cells that provide the architecture for the muscle and send important signals. And those are also the cells that, after you have a heart attack, go and make scar tissue in the heart. And the scar tissue doesn't contract at all, and that leads to heart failure. So we asked, could we get these support cells in the heart, these scar-forming cells, to turn into new muscle right where they are? So now we're not transplanting cells, we're simply asking the cells that are in the organ to completely change their fate and go from a scar-forming cell into a new muscle that would beat, integrate with the existing muscle, and actually result in greater pumping of blood from the heart to alleviate the heart failure condition. And so we were, in fact, able to find a combination of key genes that actually were essential and what nature normally used to make a heart in an embryo, that if we redeployed into these cells, were sufficient to convert the fate of these cells into beating heart cells right in the organ. And that worked we did that work first in a dish and then in mice, and with CIRM's help, we're able to do that in pigs. And ultimately, we were able to put that into the commercial sector to take it to a clinical trial. And Tenaya Therapeutics, which has licensed that technology, has now advanced that to actually be able to create a product, a therapeutic product, that can be deployed via gene therapy, in this case an adeno-associated virus, which is a vehicle to deliver these genes into the heart, and we're very hopeful they'll be able to see that all the way through to a clinical trial. If that works, then we would be able to reprogram the cells in the body to turn into new muscle, alleviate heart failure, and we're also hopeful that this same paradigm could be used in multiple other parts of the body where you have support cells that are there and could be harnessed if you just knew how to hack the code and tell them to be what you want them to be. So that's one strategy that's ongoing. And at the Gladstone Institutes, as I mentioned, we have a whole group that's also focused on neurodegenerative diseases. And one of their discoveries was that, in fact, in a certain type of Alzheimer's, in mice, what they found is that there was a very discrete area in the brain where a number of cells were lost. Now, in Alzheimer's, and you'll hear more about this from Dr. Carmichael in a moment about dementia, there are a number of cells throughout the brain that are affected. These cells in a very discrete area of the brain are sending lots and lots of signals to other cells, and they were specifically lost in an animal model of Alzheimer's. And it turned out if you can just replace those discrete cells into this discrete area of the brain, at least in mice, one was able to restore memory and learning deficits. And so that, too, has now been placed into another company here in the Bay Area called Gabaron that is trying to advance that transplant technology, cellular transplant technology, for memory loss. And so these are just a few examples of, among many others, where we finally have the ability to, whether it's cellular transplant or gene editing approaches that I didn't mention, that we can imagine a world where we no longer accept that you have disease because you've lost cells or your cells are screwed up, but rather we have the opportunity to go in there and replace those cells or correct the fundamental problem. And so I have never been more excited about the potential for us to completely change the paradigm with which we deal with human disease. And you'll hear more about that from Dr. Carmichael and then from our wonderful patient advocate, who will tell you really what that means from a patient's perspective. So thank you very much.
Thank you, Deepak. I was remiss in not mentioning that a couple years ago, Deepak was the president of ISSCR, so just further evidence of the big deal that he is. I did want to just tell one sort of story involving Shinya Yamanaka. I don't know if many of you have heard this, but right after he won the Nobel Prize, Deepak hosted an event at the Gladstone where, was it pre-Zoom? Was that Skype? What was he on that day?
He was here. He and John Gertner.
Well, no, the one that I saw, he was still in Kyoto. And anyway, whatever it was. Skype. I don't think Zoom was around yet. Anyway, so Deepak is interviewing Shinya about getting the Nobel Prize, which is obviously a fantastic accomplishment. And he says something like, and paraphrasing, so Shinya, what was it like getting that call? And Shinya, as many of you know, is very modest and understated, and he said something like, well, I was on the floor fixing our dishwasher, and my wife picks up the phone and comes over and says, honey, some guy from Sweden on the line. I got on the phone, they told me I got the prize, I thanked them very much, and I went back and finished working on the dishwasher. That's Shinya, right? Anyway, so thank you. So next, we go on to hear from Tom. Thank you.
Thank you, John. So I took this as an opportunity to approach the idea of aging, age-related diseases, and how regenerative medicine and stem cell biology might influence those processes in the brain. So how does CIRM, its support for regenerative medicine, and the emerging field of stem cell biology help us understand aging in the brain? And there's really 4 categories, and I'll weave a little bit of our work into these. The first is regenerative medicine and stem cell biology as it relates to age-related diseases. The second is as it impacts tissue aging in repair across diseases. How does the brain repair, and how does that change with age? The third is how does aging itself affect cognition, something we all dread, and what do we know about that from regenerative medicine, and how can we modify that? And the fourth and final category is what does stem cell biology offer us in terms of modeling these things? How can we study them? So the first, how does stem cell biology or regenerative medicine influence age-related diseases? There's really 3 or 4 that rise to the list there. Stroke, dementia, which we heard about, but also vascular dementia, and Parkinson's disease, in which clinical trials have now emerged, and possibly specific targets in Alzheimer's disease. So in stroke, we're past what I might call stem cells 1.0. We have some clinical trials which have initiated, some through CIRM funding that involved off-the-shelf technology at the time, as often is the case when these things finally make clinical trials, embryonic stem cell-derived neural precursors. Where the field is now is actually specialized subsets of brain cells or neurons that might have specific attributes that repair an aspect of stroke. So we heard a little bit about the kinds of cells you might transplant in a specific area in Alzheimer's disease, so-called interneurons, which are developmentally primed to home and to integrate into circuits. And there's some evidence that this might be really a subset of cells that could work in an age-related disease like stroke in transplantation. An interesting and sort of overlooked target that we've worked with is vascular dementia. So my interest in this started as a neurologist, where I had a patient who was 55 and a money manager, and she was having trouble keeping her numbers straight and kind of managing her accounts. She looked fine, she was healthy, she was only 55. I'm older than that now, and she was a frequent exerciser, but she had untreated hypertension. When we had an MRI, she had the parts of the brain that connect regions, the so-called cerebral white matter, really lit up. They were damaged, and this is what vascular dementia is. About 30% of all strokes, the second leading cause of dementia, and we now recognize that actually Alzheimer's disease exists most commonly as mixed dementia, the Alzheimer's pathology and vascular dementia. So here's an interesting sort of endophenotype, if you will, a disease within a disease, a distinct cell biology. The connecting areas of the brain, the white matter, seem to deteriorate, and there's no therapy. So I developed an animal model to study that. There really wasn't one. And to make a long story short, we progressed through to a stem cell therapy for that we've published in the last couple of years through CIRM support, which seeks to replace and stimulate the connecting areas of the brain to repair and how they ensheath and insulate and allow the brain to signal better. So there's a second example of an age-related disease in which stem cell biology or regenerative medicine might offer a therapy. There are now finally clinical trials in Parkinson's disease, which is a very prominent age-related disease we hear about, which really reflect the long-standing laboratory-based patience to take pluripotent stem cells and nudge them into the kind of cell that would be necessary in Parkinson's disease. So again, sticking at a high level in a description of the field, it's exciting to see those move into clinical trials in the US and Europe. The second category is tissue repair in general. So the brain doesn't repair well. It's not like the liver. It's not like the peripheral nerve, where we know if we injure a peripheral nerve, it will regrow back to its target. And with age, the brain regenerates even and repairs even less well. And so another area in which stem cell biology is really giving us a handle on things is age-related decreases in brain repair, particularly with the brain's own resident stem cells. And so in the human brain, the most common stem cell is a stem cell that helps repair that same area I talked about, the white matter. This is called the oligodendrocyte progenitor cell, or OPC. And so this cell exists as 10% of all cells in the brain, is multipotent, and is a tissue-resident progenitor, an immature cell that might repair. And to stick to the topic, with age, it really slows down. It doesn't divide as fast. It doesn't respond to cues. The brain itself becomes stiff and fibrotic more readily with injury and age. And so this second area of understanding how regenerative medicine can impact aging is to understand how that cell slows down with age, this progenitor or stem cell in the brain, and how the brain itself accelerates in an abnormal way at scar after injury, like stroke or in other conditions. And we're making progress understanding why the brain gets so stiff in the aged individual, and the landscape of genetic control of these progenitor cells. How might we transform them to repair better? Some of this relates to chromatin structure into epigenetics, which will be really fascinating as we start to understand control points in those facets. A third area is something that's very aspirational, and that is we know the brain slows down with age, period. Where did we park the car in the mall parking lot? Or where did I stick the key? And that's just normal aging. It's exacerbated with age-related disease. And through regenerative medicine approaches, we're starting to understand the signals that the aged brain activates that slows its cognition down. And one of these was work that I've done in collaboration with Dr. Alcino Silva at UCLA, where we identified a molecular stop signal for memory, a signal that's turned on so that memories can remain segregated. So you don't want a fuse walking into this room and sitting down with speakers 1, 2, 3, or 4 in a single, incoherent memory. We have to have junctures in which we store memories and keep them separated. And it's interesting that with age, these border zones formed in our memory are expanded, and the signals that mediate stop is increased. And this is an interesting aspect of regenerative medicine in the brain, in which we discovered these, found in some areas of injury, like diseases like stroke. These areas are these molecules are abnormally activated and impair recovery. So there's a link between what happens with age and how we slow down molecular memory stop signals. Stroke also, by accident, activating these same signals and providing a pharmacological therapy to both treat maybe the cognitive slowing of normal aging and certainly to enhance recovery in stroke. And so we moved into clinical trial with that in my lab with a large consortium in Canada, and we're working towards understanding if one of these molecular stop signals can be blocked to enhance recovery in stroke. And so the final category of regenerative medicine and how it might impact aging is in model systems. And that's something that a meeting like this is really well developed and representing. Cerebral organoids we hear a lot about, and they certainly are an interesting way to move forward in an organized association of human brain circuitry. One of the things when we think about aging that's limited this is the fact that it's a neonatal circuit, even as we let them mature for years in vitro. But one of the things that's emerging that's interesting is with some of the genetic risk factors for neurodegenerative disease, we can stress organoids in a dish and find a phenotype. Even though it's a young circuit, we can find that they don't repair their DNA as well, or they don't respond to abnormal signals like free radicals. And so this final aspect of regenerative medicine and aging is to model age-related diseases in a dish using stem cell biology in ways in which you layer on some of the stressors that we experience throughout our lives, but in which you can detect signals for and then screen small molecules and other things to impact. So that's sort of my story of the brain and aging and regenerative medicine. Thank you very much.
Thank you, Tom. Description of very, very interesting work. Now, Adrian, to present the patient advocate perspective, which is what we are at CIRM all about. So Adrian, you're up next. Thank you.
Thank you. Thank you for having me. Thank you for allowing me to come and see these beautiful young scientists. So I have to say, this is my happy place. Not that, like, you know, the really old scientists don't make me happy on that, no. But the young ones, oh my God, bring me such joy. And I want to thank you for all the sacrifices that you were making. I know how hard it is. I know that many of you are in debt. And I want to say to you that you and the work you do is not only going to change lives now, but lives in the future, okay? I want you to remember that. When it gets really, really hard, I want you to think about that. If you were a car engineer, and those are very cool and all that, and they make a new car, that's nice. But is it going to change lives and make lives better in the future? And whatever aspect of this field you go into, whether it's research, I bet many of you who've changed and gone into things like even cells, right? All of those things together create an industry that has to be in place before people like me and my family can benefit from it. So I'm just going to start off by thanking you and thanking your families, because there's days you aren't there because you're doing really, really important stuff. So just hold on to that, okay? All right, so I'm Adrian. I'm a mom. I'm 5th generation of mothers in my family to have a child born with sickle cell disease. And sickle cell is an inherited disease where it's just one gene that's miswired, and rather than red blood cells going about through the body, distributing their oxygen and making everything happy and go as it should. Once the cells release all of their oxygen, they change shape. And rather than be round and bouncy and juicy, they are sickled shape and prickly. And they break up and they release heme in the capillaries. Well, heme's not supposed to be there. That's why red blood cells are made the way they are. And so it starts inflammation. It causes great pain and tissue death. And that's everywhere in the body. So it makes it a really cool thing to study because it's, you know, it's blood and single gene. So, that part's really cool. But it affects the entire body. The pain that everybody talks about as the hallmark of the disease is simply just a signal that the body is not getting the oxygen it needs, and it's basically kind of like they're drowning for lack of oxygen and burning all at the same time. So, what brought stem cells research to it? So the idea initially was if we can turn off, right, this gene and make it not, cause the sickling, that will stop the disease, which is very cool and also very accurate, as they have proven, right? There are a couple of ways we can do that, and CIRM has funded that through vector, through CRISPR. There are lots of different ways, and you really brilliant people are going to continue to find even better ways to do that, right? But these babies are born with this. So that means from the time they're about a year old and they start going from going from fetal cells, which give them some protection, to adult cells, this process starts. So if you can imagine all through the body, the sickling's happening, it makes these clunks and it causes this pain, but that really means that there are blockages going. So if oxygen isn't getting where it's supposed to be, right, the tissue around it dies. So you think about that. So that's happening. If you have all this inflammatory response to the heme that's released, right? So now you've got that going, then that really leads to all the major organs in the body being damaged on a daily basis, right? So stroke is a very, very big deal, right? And the children can have strokes as young as 18 months. My eldest brother, who was this very beautiful, bright, bubbly child who had pain, at 3 years old had a stroke, and it left him debilitated. So he was mentally and physically debilitated. At a time when children like him were sent away, right? My mother said, no, we're going to keep him here, and he's one of us, and he's going to stay here, and we're just, wherever we go, he's going. But children like that were put away. Today, we have things like some treatments, right, that when a child has a stroke, they put them on special treatments, and they they watch them, right, and they delay that happening in many cases. And usually, if they're going to have a stroke, they'll do it probably in their 20s or something like that. Again, this damage. So there's damage to the brain. There are all these tiny, you know how the brain goes. It's got all these folds and everything. These insults that come from the blockages cause them to have many, many silent strokes. And as a mother, I remember with my daughter, because she had very great care, and we were watching her, I knew about strokes, I knew all these things, but just like one day, nothing made sense. And I kept saying, she's different, everything's different. Every time she started a car in driver's ed, she wrecked it. So, of course, being the mom I am, we had her eyes checked, we had this checked, that checked. Turns out she was having these silent strokes, right, that we didn't know about. So there's the brain, so that we have strokes. Well, there are other parts of the body are also having these insults, right? So the heart, they develop pulmonary hypertension, they have difficulty breathing, they have oxygen that they have to be on, and this is, if we're lucky, probably around the age of 29. Kidneys, again, so the kidneys have that, and bladder have that little fine lining, right? So all that's getting insults. They end up on dialysis. Infection, there's no spleen, because the spleen gets overworked, and either it has to be removed, right? Or it just disappears. As a matter of fact, that's how they found the first person with sickle cell, because he had no spleen. Any connective tissue damaged. My daughter, by the time she was 20, and we're looking at her, can't say it without being really upset, but we're looking at her x-rays, and I'm wondering why he's supposed to be showing me my kids' x-rays, and why am I looking at this x-rays for this really old person? And it was my daughter's. All the connective tissue. So any place where blood has to get through, any place there's these fine kinds of tissue in the body, blockages cause it to die. The bone marrow, which is very, very interesting, I had to learn about it. Bone marrow, which we need, so they harvest the bone marrow, right, to give them the transplant. They're using vectors and things. Our blood, their red blood cells live maybe 10 to 15 days, maybe, right? So that bone marrow is churning, and we think that's really cool, it's churning, because if you cut me, I bleed, right? But by the time they're 19 years old, the bone marrow is aging. Right? So I want my daughter cured. I want her sickle cell to end. I want her not to have pain, and you've proven that even if you replace 15 to 20% even of her sickle-shaped cells with whole blood, she will not have that terrible, terrible pain. But at 20, she's still going to have the body of a sick 60-year-old. She's still going to have trouble seeing because, of course, the retinas go. She's still going to have trouble bleeding, right? I'm sorry, not bleeding. She's going to have trouble breathing. So to me, when I went to my first World Stem Cell Summit meeting, and I could see them stopping this pain, right? But not completely ending an early death. In the state of California, people with sickle cell live 10 to 15 years shorter than any place else in the country. Then I think it's Mississippi. They have sickle cell disease. So I didn't see her really, really living a lot longer. I saw her not being in pain, but I also saw the promise of regenerative medicine. And that, to me, has to be the way we go, because to stop a disease that causes terrible, terrible side effects and damage is one thing, and it does improve the quality of life, but it does not put you into that state of wellness where you can really fully enjoy your life. And it's kind of a funny thing, because when we talk about it, we're always talking about old people, right? But I see it as a means of studying the process of disease and repairing it as a way to give a gift of life, a true life, to younger people who have suffered. And so curing is not, to me, the end of it all. Repairing, fixing, making somebody have the possibility of a whole life. A long life is really good, but a long life in pain, a long life of limits, that's really great, I guess. But the work you are doing is going to make a life without pain a life with possibilities. And if you've ever had a life where as day to day you find yourself being even more limited, as people with sickle cell do, and a lot of other illnesses. The idea of having the gift of doing even simple things, skipping, running, driving a car, having a job, having children, things that people who are healthy sometimes just like breathing take for granted. Being able to give that to people who have suffered. Generations of my family have suffered and died, right? I have a child that I watch suffer. But since I have found out about your work, I don't carry that grief anymore because I know that the work I do and the work you do and our partnership is going to stop that, not just for my family, not for the 100,000 people in the US who have sickle cell, not just for the millions in the world who live for sickle cell, but all of the diseases. And so that's where I see us going. Right now we talk about aging. I think of it as living, and I think of it as something that is our duty, because as we learn more and know more, it is not only the really cool, wonderful thing, but it's also the humane thing. And so the work we're starting, the work we're doing, the work you're going to do all together, a brave new world. So thank you for listening. Thank you.
I think you can see why when we say Adrian is one of our shining examples of a patient advocate who makes a real difference and drives us to do what we do. That was wonderful. Thank you very much. Should tell you that CIRM, as Adrian noted, is funding a number of projects in sickle cell, both CIRM as CIRM, and we also have a collaboration with NHLBI where we are jointly funding sickle cell projects, and the cure for that is going to come. It's not that far out. It is really exciting stuff. Okay, so we're going to turn to Q&A, and are there any people in the audience that have questions they'd like to ask? And if you would, there are microphones scattered around, and I'm going to, in the meantime, while people may be going to the microphones, ask a couple myself. Now, one of the interesting things about regenerative medicine is it's not just cutting edge, but there's some pretty wild stuff going on that you've probably read about out there in the field. And I wanted to ask our panelists about a couple of them that are, I think, particularly wild. So Deepak, parabiosis. Could you discuss what that is for the audience here and give a few comments on the promise that you see that that may have in the aging field? This is wild stuff.
Yeah, John, parabiosis is pretty wild, and the notion in this, for those of you who may not be familiar with it, is that there may be some factors in a young person or animal's blood that, when transferred into an older individual, rejuvenates and makes the older individual's cells younger again. And it sounds, science fiction-y or just wacky, but in fact, you can see if you sew two mice together, which has been done, and their blood mixes, then if you sew a young mouse together with an old mouse, a number of features of older organs that we can measure are clearly, those organs now look more similar to a younger organ. So that I think there's tremendous promise in that line of investigation, obviously not in sewing people together. The older person might not mind. But if we could figure out what is circulating in the blood of the younger individual and purify that and turn that into a therapeutic, then I think that could be given to an older individual. But what it does say, which I think this is the most important part of that line of investigation to me, is that it says that this notion of a cell just getting old is reversible. You just think about that. That it has to say that. And that wasn't, I don't think previously was not obvious, and still is not obvious to me how that could happen. But the fact when we observe that, I think one has to step back and say, the idea that an old cell can't go back in time is just wrong. And it's just up to us to figure out how to do it.
Thank you. Pretty cool stuff. Another thing, sort of ripping from the headlines, for you Law and Order fans in the audience, you read about a couple months ago a transplanted pig's heart into a patient who was terminally ill that had involved, it wasn't just a pig's heart, it was a genetically modified pig's heart, which had 10 different genes that were either knocked out or knocked in and combined, one of which was pretty interesting, which was the knockout of the gene that controls the growth of the pig's heart, because the pig's heart left to its own devices would get to be too big for a human transplant. Anyway, it had all these different genetic modifications, and they had the transplant. The patient lived for a couple of months. I'm not sure they've yet figured out exactly what happened that caused the patient's death, but they don't believe it was the transplant or the functioning of the pig's heart. So Tom, could you just comment on that? I'd be curious to hear what you think of that and this whole notion of modified animal organs as a potential for transplant in humans, which of course, if ever came to fruition in a major way, would address the serious organ shortage that we have, folks with degenerated organs of one kind or another.
It's certainly very exciting. It's certainly not going to happen to the brain. So I'll wander a bit in my organ-specific focus. But one of the things it heralds is a moment of time in which we're now past the conceptual stage, and now we're now into tuning. We're into iterative science. I think that was one of the exciting things with that. We know, we've seen a lot in this meeting, we know it from the literature, that there are various ways to move forward, such as a universal cell sort of approach, which is might be immune, invisible, and that might be a way to do it. Another way is to iteratively manipulate genes that allow an organ to maybe not be totally transparent, but in a more easily done way, be integrated with less immunosuppression. And so what it says is there's several pathways forward in a critical field of shortage for transplant, and we're now in the stage in which we can start to tune these. We can do so more easily with genetics. We can do so in the universal approach by tuning a handful of genes. We can do so in the pig approach by trying to tune more of them. But it's now that stage of the field in which we can tinker and move forward rather than have this big conceptual block.
Deepak, do you have any thoughts on that one? This is a little bit more in your...
Yeah, I followed that closely because there's a heart transplant. As I mentioned earlier, there are not that many heart transplants done worldwide, and it's largely because of the shortage of the organs for donation. So I thought that was simply a phenomenal result, and it was actually after decades of trying to do this, and it was a very unique situation in which this person wasn't going to get a human heart transplant, couldn't be on the list, and so it was that do nothing or die, and so it became ethical to at least try this pig heart in the patient. But it was after decades of effort as trying to find all those key things to mutate and change so that our body wouldn't reject the pig heart. And it's nothing short of phenomenal that worked. And you're right, the patient didn't apparently die from rejection of this. There's some other issue. And so in my mind, that was 2 months, living 2 months like that. It was a huge success. And I think we'll see more and more of that. So I'm very excited about that work.
Thank you. And I should add, the same company that did the procedure and developed that, the pig heart, also had a pig kidney that they used in a different way. Instead of transplanting that into a normal patient because they were concerned about a number of things, they took a patient who was basically brain dead on life support and hooked this transplanted kidney to that person to see if it would function as a normal kidney, and sure enough, it did. And so another example, this is the sort of, in the field of organ transplants, this is one of the waves of the future. Adrian, I want to shift gears completely. And I'm just curious, as you kindly said, CIRM in California are doing their part in terms of funding for sickle cell research. What is your view of the current state, and NHLBI as well, but your feeling of the current state of support for sickle cell research, but beyond that, just research in general at a national level?
You've heard me say that I'm living my best lifetime, in my sci-fi life. Sickle cell was a known disease for 125 years, and we went 110 years of not really having medicine, a treatment, other than protocols to fight infection, right, and blood transfusions. And we finally got, in the last 3 years, we got 3 new treatments. But most of the work that was done to sickle cell prior to this point had only to do with pain, right? And so CIRM came out in 2016, I think, 2016, and was with the largest grant given for sickle cell research in many, many years. And it sort of caught everybody's attention. They were like, oh, okay, sickle cell, money, research. And it was like started a firestorm for us in that we got so much attention, and then we were able to go to the government and say, you know, this needs to be looked at. This isn't fair. We got training in things like communication, presenting, and as a result of some very strange things coming together, right? So the money issue really, CIRM started that, I have to say. They did. Nobody gave us lots of money. Although there are 100,000 of us, and we work very closely with the cystic fibrosis group. Cystic fibrosis has I think, a tenth of the amount of patients we have, or some number like that, but they get something like 300 times the funding. So in the last 10 years, we have gotten funding from the government. We have gotten, and I want to say at the federal level, at the state level, in the past 5 years, they've given us close to $20 million for clinics, which we never had. Again, because CIRM highlighted the need for this. And we as advocates could go and say, look, California, you're giving us a cure, but what about treatment? I mean, we're dying. And so we're kind of at this sweet spot. We are actually going to get a registry. And California is leading. Also, 2 of the treatments, the 3 treatments that were created were done here in the state of California. So again, that brain trust that was seeded by CIRM has really, really paid off for us. And now our fight is to get, good care when we actually go to the hospital, which, and we're freed up to do that because we don't have to fight anymore for research, and we don't have to fight anymore for anybody to think about creating any treatments for us, right? That used to be so overwhelming, that the idea of just keeping our people alive and then the hope of getting something done. So we are really at a very sweet spot, brought about by a lot of pain. Also the DEI discussions going around now. Because before we never could, we would go and we would talk about the inequities. And we would say it's not really inequities, we're just not getting what we need. It is not equal. And much of our time speaking would be trying to explain to people what that looked like, right? Being people of color and not having, there's a saying in Asia of same. Well, we didn't have the same, and we would have to explain this to people that we didn't have the same thing, and that the conversations now are more of why not, and aha, I see it. So I think I always said that, we were kind of on the edge of many things, but I think we're on the edge of this also, right?
Thank you. Yes, so we have a question back here from the audience.
Okay, thank you. I have actually a comment and a question. The first relates to what Deepak was talking about, heterochronic parabiosis. Indeed, it's very fascinating, and we don't quite understand what pathways and ligands are activated when you join the circulatory system. But this year, there's a paper, actually, where they sequence single-cell profiling, RNA-seq, before and after the parabiosis. And I think it's a very interesting paper that we can use that data to interrogate which kind of cues are actually coming from the young mice to the old, and how this rejuvenation, let's say, happens. Now, my question is related, but not completely.
Could you just speak a little bit closer to the mic? I'm having a little bit harder time hearing.
Can you hear me better?
Yeah,
now?
yes,
Okay.
thank you.
So my question, which is related, is the following. When we talk about cell transplantation, we usually focus on one cell type: Parkinson's, dopaminergic neurons, any other cell type, beta cells. But do you think it's a challenge that we have to address, that not only we should transplant specific cell types, but also think about how to reprogram the niche, since we don't even understand what are the causes for degeneration in the first place? And let's say disease niche could also be influencing the degeneration and persist in this case.
Tom, do you want to take that one?
That's a great question. So in the brain, it's really all about the particular niche of disease or signaling. And so in what I talked about, it's sort of a neurovascular niche in white matter, the myelinated areas of the brain. In other diseases, it's more of a niche that's in a particular subset of the brain. In Parkinson's, they're putting things into the basal ganglia or the putamen. But I think the idea that you have a cell-cell signaling environment that transmits both health and stress, and you have to pay attention to it and probably put your cells in the middle of changing that, I think is a key thing. And so, for example, in the white matter, the progenitor, it's really a progenitor cell transplantation that has gotten most traction, a glial progenitor that doesn't go in and become one specific cell type, but it modifies the neurovascular niche of the white matter. So I think you're right on. You're not putting some sort of kind of drone in that kind of goes in with its propellers and does a pre-programmed flight. It's instead going into an environment in which it's changing how the cells signal to each other.
Another question.
Yeah, I'm Al-Hindi, MEG surgeon at University of Chicago. So Adrian and everybody, great talks. Thank you so much. So your comment about the cystic fibrosis and the sickle cell and the level of support, obviously a flagrant example of the health disparity, which we see in many diseases. Many of the diseases I work on in women health, also clear examples of health disparity. I was wondering if you just want to comment on the status. You alluded a little bit to that, but if you want to elaborate more on the state of the art on this, what the NIH is doing and so on. There's a lot of talk, but I don't see a lot of practical steps on this. Thank you.
Okay, watch me so I don't go over. Okay. So yeah, that's interesting. We are working very closely with NIH and the FDA and ASH, which is the Association for Hematologists, and many researchers. And they're always coming to us and saying, this is what they say, well, we can't get you to come in and sign up for clinical trials. You know, black people and brown people, they just don't trust science. And so we did a bunch of surveys, right? Because we were saying, tell us what kind of data do you need from us? And they were like, well, you know, we just need numbers. So we did over 500 people, which is in a very short period of time. And we were like, so what is it about clinical trials and research don't you believe? Now, there were all these hypotheses that they were going to come back and say, well, it was the Tuskegee, it was this. But you know what it came back to? I am treated so badly at the hospital, at the clinic. If I go and I do a clinical trial, what is going to make them treat me any differently? And if something goes wrong and I have to go back into the healthcare system, what's going to happen to me? That really is it. So it is a thing where we as people of color, and even I think to some degree whites who are poor and uneducated, have the same kind of thing happen to them, right? Where you're not offered the best care, you're not given the best care. And I just had a simple example of that, where you're not, we sent my daughter in, and there were to get her bone spur repaired. And the list of things that was offered to her was a subset of the list of things that everybody else got, right? And it was assumed that because we might have to pay for it, right? And couldn't because they didn't know who we were or anything, they didn't even offer her to it. So that really is it, our experience, and it's intergenerational, right? So it's not a mother going and taking a child and saying that my child was treated badly. The mother is going and saying, oh God, I know they treat me badly, they taught my mother badly. So until we can improve the quality of care that people receive, we're not going to be able to really gain the trust in new things. Now, what CIRM has done, can I talk about CIRM?
Please.
Okay, so what CIRM has done is they have, I want to say embedded, but they have included patient or disease advocates in the clinical trials. And I can talk about my experience as part of that and some of the others. But we are giving parity when we're having these discussions with the scientific team. And so we're able to sometimes, more often than not, we voice what needs to happen, and then they make it happen. For example, said, you know, you've got to understand the culture, right? You've got to understand the financial situation. For example, UCLA charges, is it $15 a day for parking? $15 a day for parking. It's on the west side of the city where it's the average place for living is what? I mean, an average house now is $1 million, so probably $3 million.
It takes a lot of petrol to get from the other part of the city there. It takes a long trip. And so I'm like, why are you're asking them to drive? You're assuming that they've got a car that's good enough to make it, right? You're asking them to pay for petrol. You're asking them to pay $15 for parking, and you've designed it for a family that's already stressed and probably an hourly wage earner who is being the support person, and you want them to come 3 days a week. That just, and they were like, oh, well, and one guy said it's only $15. And I had to say, do you realize that if you have sickle cell, you cannot work, you are too sick. Nobody's going to hire you. And if you're the caretaker provider, you can't work because they want you in the seat. I had another word for it. But if you're not in the seat, you're not earning money. So it's always constantly this triage. And the concept of that really surprised them. It surprised me that it surprised them. And so, and I was like, and even the cultural things, they were like, well, this patient came in with her mother. I said, was it really her mother? Was it her godmother? Was it her auntie? Was it really her mother? When you do an assessment of this, do you understand the thing about, silent strokes? So we were able to put things in place, right? Work with my organization and other organizations to provide things like transportation, to provide food. Because if you're there all day, you need to eat. Well, it costs a lot of money to eat at UCLA, I'm just saying. No, really,
but
seriously.
at least it's 5-star cuisine.
It is. Now if I could get the hospitals to listen to me when I go, I want a hospital. I don't want a 5-star hotel. You could use this money to do something other than, at any rate. So as we design the trial and have people come in and say, well, we can't pay for these things, but we have an organization here that's going to do in place to do these things, to support you, right? It was amazing. It made such a difference in the patients really saying, okay, I have someone here. I have a place here that understands my challenges without making me feel bad. They didn't say how much do they make. They just said, we are going to provide this for you. And so we were able to get to more patients. But really, truthfully, until we improve what happens at the state or the point of care, it's going to be an uphill battle.
Kevin, are you asking a question?
I'm asking a question on behalf of one of the people online. And this was Nancy, who asked, what about the effects of the environment on DNA? How do things like pollution, dirty air, and water affect aging and lifespan?
Yeah, it's an important point in terms of the number of environmental influences that could affect our epigenetics. Maybe some can even affect our DNA code, but more often they're affecting the epigenetics, or how the DNA is actually read. And there are a number of examples of environmental influences that do that, and thereby influence aging. And so I think we now have the tools to actually interrogate ourselves to ask how various environmental influences are affecting our epigenetics, which we didn't have before. And I think those will be very important lines of investigation, because then once we understand that, we can try to either stop the environmental influences, or if we can't stop the environmental influence, which is often the case, we can at least mitigate the consequences if we know which cells it's affecting and how. One example we're doing right now in our laboratory is trying to understand how maternal diabetes affects the fetus. And we know that children are born with a higher instance of cardiac and craniofacial defects if the mother has high glucose levels. And we've never known why. And through modern technologies that we didn't even have a few years ago, now we've, for the first time, understood that there are very selective cells that are vulnerable epigenetically to be completely altered. And that's likely the basis for these defects. And that can be done now for most environmental insults that affect aging, so that will happen.
So I've got a question for the panel here. What, on the controversial topic again, was just reading very recently about a stem cell clinic in California that was promoting anti-aging techniques that were going to fundamentally alter your life and turn you back to a teenager. Yeah, sign Deepak up. There are many of these predatory clinics out there. Could you guys just comment on that and what you think can be done to sort of make them go away, which is going to be virtually impossible?
Well, okay, I'm going to go first because I live where I live. So I live in LA, but I'm in this little tiny valley called Eagle Rock, and on one side is Pasadena, on the other side is Glendale, and it's filled literally with like beauty spots, right? And they have all of these things where you can go in and, you know, you can get, I don't know, fat melted and eyelashes and all these really great things. But they also have a section where you can go and you can get stem cells to get hair. You can get stem cells injected in your knees. You can get, and so, you know, I knock on the door and I go, you know, where's your proof, right? And the operators they don't have any proof. They have these kind of made up studies. So I say to people all the time, in everything, we find people who make promises to you. But bad, cheap science, and when I say cheap, cheap operational, cheap not as in not studied well, not a clinical trials, those kind of things that cost a lot of money, and people don't really understand why it costs a lot of money. So we have to educate them more. And I report the businesses, right, to the Better Business Bureau, but I don't know that there's anything they can really do. We have to educate people because they're being harmed and are being robbed, are being harmed and robbed. Yeah.
So this has been a big focus of the ISSCR for some time because globally this is an issue where there are a number of rogue stem cell clinics that will promise everything under the sun that can be cured with their quote-unquote stem cells. But it's been a difficult problem, and at one point we tried to actually indicate that a list of places that we thought were not good stem cell clinics, were not validated information, and from a legal standpoint we ran into trouble. I see George Daly in the back of the room walked in, and he was, I think, very involved with that also when he was president of the ISSCR. What we ultimately resorted to was an alternative was to simply try our best to educate the public on what questions to ask. So we could empower the public to try to get the information they needed and empower them to how to interpret that information and come to their own conclusion about whether there's sufficient evidence of whether or not the therapy promised by the clinic actually could work. And so we created a website that would have all the information, the questions to ask, a list of those, and what kind of answers they should be looking for to get comfortable that this might be worth their $20,000, $30,000, $50,000 or not. So.
Thank you. Tom, we have 3 minutes left, and I wanted to ask you about another controversial topic that is brain-related, which is the FDA approval of Aduhelm and the controversy surrounding that, and what does that say about the FDA review process, and how can you extrapolate that into how is the FDA going to approach evaluation of difficult new treatments like cellular therapies, et cetera?
Yeah, that's a great question and probably a political minefield. But there are a couple of pressure points and lessons that came out of that. One, I think, is under current investigatory review, and that is, was there some sort of internal set of communications and pressure in the FDA from the pharmaceutical industry that changed things? And I think we're waiting to see what that report shows. But but in general, I think what it shows is you've really got to follow the science, and that there is a progression of evidence that moves forward that we all know in this room, and the public will know this too, particular phases of clinical trials and the kind of results we get from biomarkers or from the patient that's laid out, that indicates a therapy has efficacy. And I think we just need to make sure that we understand those, that there's a roadmap, and that we follow it. And then when the experts feel that it has or has not been followed, that the regulatory agency listens to the experts, and we don't have some sort of exceptional divergence.
How far do you think we are from a viable treatment for Alzheimer's of any kind?
Well, there's aducanumab, which came out, and it had an effect that was very modest in one way the data was analyzed. There's 2 more antibodies against various versions of amyloid that are following that progression that look like they may have also a very modest disease-modifying effect. So I think that starts to turn the field to say, now how can we capitalize on that? I think it is a paradigm shift, but it's very modest, and it puts us in the position of amplifying that rather than having to start from scratch.
Great. Well, we've now reached the end of our hour and a half.
JT, there's a lady who's been waiting to ask a question over there.
Yes, please.
Yeah, I'm just struck by the extraordinary need that there is, and how I think the FDA is a science-based organization, but when they have an advisory committee meeting, there's like the audience is filled with patients and patients' parents who are doing badly, such as with Duchenne muscular dystrophy. So there's extraordinary pressure, and the question is, what is good enough, and what the benefits and risks are. I mean, it's extraordinarily difficult. And I wanted to mention that one of the posters talked about some kind of Medical Freedom Act that's been passed in several states, where people say they have a right to stem cell interventions in particular. And I was wondering if there's anything that society or CIRM is doing about that. It's sort of, again, it's sort of a right to try kind of thing that can be sort of anti-science and in a way harm patients in the end.
Well, I'm going to say this. I think that the FDA, for a long time in many ways, was underfunded. I think that if we as advocates who have been waiting for these treatments and we're saying, oh my God, this was fast, imagine what it was or is like for the FDA to try to get, as a government agency, to get everything staffed up, educated, and work this out. So I think that part of it is that they are working it out. And I know that we as advocates go, many, many desperate. I mean, stem cell treatment, tourism is alive and well. And that's generally because people are so desperate. So I think we're looking and saying, how are we all, I think we all together have to work on this problem. And I don't think we can point fingers at the FDA or at the advocates or the companies producing it, but we understand that it is a moving target. And because it's a technology and a new way to do things, you know what worked and what didn't. And it has to be, rather than going through a whole long process and then coming back and refining it, we have to have shorter intervals of our evaluation and shorter steps of looking at things and making it faster to be able to implement what we learn. I think that that's what needs to happen.
Okay, well, with that admonition, I think that's a good place to stop. I'd like to thank our panelists very much, and thank you all for attending, and I hope this was an interesting session.
They are. I should not have been queuing up.
No, it's okay.
Oh, okay.
It's perfect that you did.